Hello everyone, welcome to our series in which we are covering the entire syllabus of ASRB NET Agriculture Entomology. So, in the previous video we had covered the body wall of the insect. So, in this video we are going to cover the important questions regarding body wall of the insect. So, in epicuticle, chitin is absent. Chitin is a polysaccharide which is absent in epicuticle. Second question. The thickness of the epicuticle varies from 1 micron to 4 micron. It is simple written that the thickness of the epicuticle is 1 micron to 4 micron. Third, procuticle is differentiated into exocuticle and epicuticle. So, we had studied the cuticle portion. The cuticle is divided into procuticle and epicuticle. So, the procuticle is divided into two regions, exocuticle and epicuticle. The epicuticle is the outermost part of the insect cuticle. Fourth, procuticle contains chitin and protein complex. So the procuticle contains both polysaccharide and protein complex. Sclerotin is predominant in exocuticle, whereas arthropodine is predominant in endocuticle. So here we see that sclerotin is predominant, means in major quantity in exocuticle sclerotin is present, whereas in endocuticle arthropodine is present. Sixth, sclerotin is water soluble insoluble tanned protein. So sclerotin is a water insoluble protein and arthropodine is a water soluble protein. Eighth, arthropodine consists 25 to 37% of the cuticle. Arthropodine is a protein, we all know that, and it makes 25 to 37% of the cuticle. Ninth, tanning is the process where arthropodine is converted into sclerotin. So it is the process in which arthropodine protein is converted into sclerotin. 10th, sclerotin and tanning is due to quinones derived from tyrosine. The hormone required is bursicon. So स्क्लेरोटाइजेशन एंड टैनिंग जो प्रोसेस होती है उसमें आपका बुर्सिकॉन जो हार्मोन होता है वो रिक्वायर्ड होता है. 11th, एपिडर्मिस और हाइपोडर्मिस इज अ सिंगल कॉन्टिन्यूअस लेयर ऑफ सेल. So epidermis is a single continuous layer of the cells, means it's not multi-layered, it consists of only a single layer. 12th, adjacent epidermal cells are connected to each other by cytoplasmic processes known as desmosomes. So the connection between the polygonal cells of the epidermis is by the cytoplasmic processes which is known as desmosomes. 13th, all the epidermal cells are granular and secrete cuticle and enzymes involved in the production and digestion of old cuticle during molting. So epidermal cells are granular and secrete enzymes that are involved in both formation and digestion of the cuticle. 14th, dermal glands are modified. Epidermal cells produce cement layer. 15th, epidermal cells secrete wax layer. 16th, oinocytes produce lipoprotein which are deposited by epidermal cells to form protein epicuticle. So here we are saying that oinocytes produce lipoprotein which are deposited by epidermal cells to form protein epicuticle. The epicuticle is formed by oinocytes because it produces lipoprotein. Epidermal cells secretes cuticle. 17th, epidermal cells secrete cuticle. 18th, basement layer is a non-cellular membrane underlying the epidermal cells of the integument. So here we are talking about basement layer. It is a non-cellular layer and it is underlying the epidermal cells. We had studied in the video also that the basement layer is a sheath-like structure just below the epidermal cells. Basement membrane is 0.5 micron thick. 20th question. Chitin constitutes 30 to 40% of insect cuticle. Means chitin, which is a polysaccharide, it constitutes of 30 to 40% of the insect cuticle. 21. Chitin is abundant in endocuticle. So in endocuticle, here means that the chitin is present in a big quantity, large quantity in endocuticle. Chitin is chemically C8H13O5 N whole X. Specific gravity of chitin is 1.4. Chitin stretchability is 13 mm. Alpha chitin is the most stable component present in arthropods. So alpha chitin is the most stable component present in arthropods. 26. Presence of insect contaminants in food material is known as Chottian test or Van Wesseling test. 27. Shading of old cuticle accompanied by the formation of the new cuticle is called molting or ecdysis. So it means that where the process where the prior layer of the insect is dissolved and the new layer is formed, it's called molting. Detachment of the cuticle from the epidermis is apolysis. So it means that when the body is when the cuticle is detaching from the body of the insect, it is called apolysis. The time interval between two molts is called stadium. The form of an insect during stadium is called instar. Means the phase of the insect between the two during the stadium is called instar. The next 31 is the cast of skin by an insect is called exuvia. The skin which is shaded, here we are talking about the skin which is shaded by the insect, is called exuvia. 32. Molting fluid contains enzymes which is chitin and protease. So here we are talking about the molting fluid which helps in the molting of the insect. It contains of chitin and protease. 33. Schemat's layer is the zone of deposition of new cuticle or procuticle. So we are talking about here a layer or a place where the new cuticle is deposited. 34. Order of deposition of the cuticle is cuticular layer, procuticle, wax layer, cement layer. Here might be incorrect that curriculum layer. It should not be curriculum layer. It should be cuticular layer. A cuticuline layer. 35. Verson's gland is the molting gland of caterpillars. Means Verson gland is the produces molting fluids in the caterpillar. Teneral is the new emerged pale soft-bodied individual prior to the completion of melanization and sclerotization. In the upcoming questions, we will know about what is sclerotization and melanization is. So here you just have to understand that teneral is the newly emerged pale soft-bodied individuals prior to completion of the melanization and sclerotization. So melanization और sclerotization से पहले जो soft body वाले insects होते हैं उसे teneral कहते हैं. 37. The elastic protein responsible for the flexibility of the wing sclerites is resilient. जो flexibility पाई जाती है insects में, insect के wings में, वो किस protein की वजह से होती है? Resilient की वजह से. 38. The process by which cuticle becomes hard is called sclerotization. So जो आपका cuticle कड़ा हो जाता है, hard हो जाता है, वो किसकी वजह से होता है? Sclerotization की वजह से. And the process by which cuticle becomes dark is called melanization. और जो उसका डार्क कलर होता है वो melanization की वजह से होता है. Maximum sclerotization is found in mesonotum of the adult insects and mandibles of larva. So maximum sclerotization कहाँ पाया जाता है आपका? Adults के mesonotum में और mandibles of larva में. Polymerization of the cuticle is characterized by the enzymes chitin synthetase. जो पॉलीमराइजेशन प्रोसेस है क्यूटिकल का वो किसके द्वारा तेज किया जाता है, कैटेलाइज होता है? एंजाइम काइटिन सिंथेटेज के द्वारा. 42. क्यूटिकुलर अपेंडेजेस आर यूनिसेलुलर और मल्टीसेलुलर इन ओरिजिन एंड बेसल आर्टिकुलेशन इज प्रेजेंट. जो आपके क्यूटिकुलर अपेंडेजेस होते हैं वो दो तरह के होते हैं, या तो वो यूनिसेलुलर होंगे या तो मल्टी सेलुलर होंगे. जो यूनिसेलुलर होते हैं उनके एग्जांपल्स होते हैं सीटे और माइक्रोट्रेकिया, और मल्टी सेलुलर का एग्जांपल होता है स्पर. 45. सीटे आर ऑल्सो कॉल्ड माइक्रोट्रेकिया एंड अराइज फ्रॉम द सिंगल हाइपोडर्मिस सेल कॉल्ड ट्राइकोजन सेल. जो ट्राइकोजन सेल है उससे आपका सीटे प्रोड्यूस होता है. द कवरिंग सेल विच फॉर्म्स द आर्टिकुलर मेम्ब्रेन एट द बेस ऑफ द सीटे इज कॉल्ड टर्मोजन. So when you will study about epidermis, you will study about these two cells, the termogen cells and tricogen cells. 46. Chitotoxy is the arrangement of bristles on an insect based on their position and size. Important in taxonomic studies. So chitotoxy is the arrangement of bristles on an insect based on the position and size. 47. Spurs are cuticular appendages, multicellular, mobile, and they occur in legs of many insects. So spur is a cuticular appendages which is multicellular, mobile, and they occur in legs of the many insects. 48. Cuticular processes are non-cellular in origin. They lack basal articulation. So cuticle processes are non-cellular in origin and they lack basal articulation. Microtrachea or fixed hairs or oculi are present in the wings of the copters and diptera. 50. Spine thorns-like form is horns larger projections. Like they are pointed thorn-like horn-like larger projections, and male of certain coleopteras. These are found in male of certain coleoptera. So thank you for today. These are the 50 important questions for from the body wall. And like, share, subscribe to our channel. Thank you for today. This is all up to this. The 50 important questions of body wall has been covered. And please like, share and subscribe to our channel. And if you are a true agriculturist or entomologist and preparing for ASRB NET Agriculture Entomology examination, please subscribe to our channel, because we are posting daily videos, daily important videos, and we are taking a series in which we are covering the entire syllabus of agriculture entomology and ASRB NET Agriculture Entomology exam. So please subscribe to our channel and share to all your friends who are preparing for ASRB NET Agriculture Entomology examination. Thank you.